All right, everyone, welcome back. Today we enter the era where astrobiology truly became a discipline, not just a dream or philosophical curiosity. Honestly, if you were a scientist in the 90s, astrobiology felt like a science fiction. But from the year 200, 2000 and onward, everything changed so quickly that even researchers inside NASA couldn't keep up. So today's lecture is about how we went from wondering, are we alone, to actually building telescopes, robots, and even synthetic life to find out. NASA Astrobiological Institute. Imagine a hallway debate in the late 90s. NASA brought together a group of scientists in a building in California. One is a geologist, a molecular biologist, an astronomer, and a chemist were arguing in the hallway about how life began. At some point, someone said, look, all of us are trying to solve the same puzzle. Why are we working separately? This sounds trivial today, but in the 90s, these disciplines barely communicated. And the conversation was one of the seeds that formed the NASA Astrobiology Institute in the year 2000. Suddenly, astrobiology had a budget, organized research notes, had graduate programs, had missions planned around these questions. For the first time in human history, a government space agency officially declared finding life is one of our scientific priorities. That alone is extraordinary. Mars had a second chance. Now let's enter everyone's favorite chapter, Mars. I always like to say it's like if you turn astrobiology into a television series or something, Mars would be the recurring character. So let's talk about it. So there's LH, the meteorite that started a war, literally. So this meteorite in 1984, a group of geologists in Antarctica found a small greenish rock, and in 1996, NASA announced we may have found fossilized life inside it. This was the Martian meteorite that crashed Antarctic region. It contained tiny magnetite crystals arranged in chains. Some look similar to magnetite produced by bacteria on Earth, and there were polycyclic aromatic hydrocarbons. The shapes inside the rock looked cell like. So Bill Clinton even gave a press conference about it, and sales of astrobiology books doubled within a month after this announcement. Was it life? Well, probably not. Today we think the structures were abiotic. But this meteorite forced the world to take astrobiology seriously. And in science, sometimes a controversial discovery is more transformative than a correct one. So, we're gonna see some rovers. Let's talk about the spirit and opportunity here. The twin brothers who refused to die. Um, these rovers were supposed to last 90 days. Spirit lasted 6 years, and opportunity lasted 15 years. Opie survived dust storms so dark the rover went into sleep mode, hoping the sun would return. And what did they discover? Rounded pebbles shaped by ancient rivers, hematite blueberries formed in water, minerals that require long standing wet conditions. By the time Opie sent its last message, which was, I urge you to go look for it, very dramatic. My battery is low and it's getting dark before it went dark. We already knew Mars once had lakes, rivers, and maybe oceans. And let's go back to another rover, the Curiosity, the methane detective. So, Curiosity rolled into Gale Crater in 2012 and discovered organics in 3 billion year old mudstone seasonal methane cycles. Now, methane on Mars is like a drum on Twitter. Just one whiff and everyone freaks out. So, possible explanations were geology, serpentinization of subsurface microbes. Well, we still don't know, but the strange seasonality keeps the mystery alive. And if you go through another important robot, the preservance. So this is really important because it's also very recent, actually. And Jezero Crater's delta is one of the best preserved ancient lake systems in solar system, and Preservance has detected organics with Sherlock technology and imaged carbon-rich rocks and selected samples for the Mars sample return mission. When those samples arrive on Earth, probably in 2035ish, they may rewrite history, and one grain of Mars mud could answer a 4 billion year old question. So, what Preservance has found in the Jezero crater, um, sort of a leopard spot, they called it, by mineralize and without explanation that can't occurring in abiotically, and maybe there was life back in the day in the under those lakes, and there were very similar systems on Earth. Let's continue. Let's talk about extremophiles and limits of life. So this is one of my favorite parts because I really into microbiology. The Earth's extremophiles are more alien than any sci-fi creature. We can say microbes that were thermophiles living at 122 degrees. Water near boiling isn't extreme to them. It's home. There is Deinococcus radiodurans, a conon the bacterium who can survive 5,000 radiation, vacuum, and extreme dehydration. It was first discovered because a spoiled can of meat wouldn't stay sterilized. And there are acidophils. Some live at pH 0, basically inside the liquid acid. Very, very interesting. There's halophiles, microbes that prefer salt concentrations higher than the Dead Sea. So whenever you see these creatures, you understand why astrobiologists suddenly said, hold on, Mars may not be as dead as we thought, and Europa, Titan, Enceladus, these aren't crazy ideas anymore. Extremophiles expanded the borders of our imagination. Let's continue the exoplanet explosion. So we're going to talk about Kepler test, TRAPPIST-1 system, and James Webb telescope. This era deserves a movie, really. Before the mid 19th, we had a zero confirmed exoplanets. Today we have over 5,000, right? So the Kepler, for example, the planet census stared at 150,000 stars and found gas giants, super Earths, lava worlds, water worlds, planets with a years shorter than a day. Kepler proved something revolutionary. Planets are else everywhere. And then we had TESS. The nearby Hunt test finds planet around stars close to Earth. Planets we can actually study. These will be the first worlds where we can look for oxygen, water vapor, methane, and more. And we have actually the TRAPPIST system here. So the system is unbelievable with seven rocky planets, three inhabitable zone, and all similar size to Earth. Even better, they orbit a dim star, so their atmosphere are easy to study. So very interesting system. And if you go back to James Webb Telescope here, the atmosphere reader is a telescope astrobiologists dreamed about for 40 years. It has already detected carbon dioxide on exoplanets, water vapor, methane, strange photochemical hazes, and the next step will be the biosignatures, which would be another subject that we will go into, but um, very problematic subject. So, what is a biosignature? So, biosignature is the idea that we decide what to look for in other planets with our telescopes to determine is there life or not. But it's really hard because first we need to know what to look for, and for that we need to understand what is life, the question that has no answer. We need to confront a philosophical problem here. We don't actually have a universal definition of life. This is surprising and difficult. Every definition you find tends to break When we push it to extremes, life metabolizes as well. Viruses don't metabolize. And or you can say even chemical systems abiotically without any life influence there can metabolize. Life evolves, but well, abiotic systems evolves too. They showed that in the paper I mentioned before, in fatty acids where they evolve, and chemistry can evolve and change the environment, and reproduction can be made artificially, not in the way we hoped. Well, cellularity, well, there might be life without cells, or perhaps ice, the ocean, host chemical networks without membranes. So life is the how Carl Sagan mentioned the exception we don't yet understand. So to determine what life is, we need a comparison. We need another example. So every chemical process, every uh, production of any chemical can also make artificially, and this is a problem. We would never know what caused it, or even the methane. You can say an organism can make it. Well, it can make abiotically. How do we determine that? And our technology is not developed enough to determine these biosignatures. With our telescopes is not enough. We need to send people there, which will create another issue we call planetary protection. We talk about that before later. So NASA has a working definition for life. Life is a self-sustaining chemical system capable of Darwinian evolution. It's not perfect, but gives us a framework when designing missions. The deeper problem, we only know one example of life, Earth life. So categories would be DNA, protein, lipids, water solvent, carbon-based chemistry. If you only have one organism in your dataset, how do you generalize to the universe? That's why astrobiology often asks not what is life, but what does life do? How does life behave as a system? So how do we search for something we can define? So life has patterns. Instead of asking, is it life? We ask, is the system far from equilibrium? Life constantly uses energy to stay organized. Is there a chemical complexity? Life tends to use highly specific, unlike molecules. Are there signs of information flow? Some patterns of storage and function like heredity? Does the environment show cycles that cannot be explained by geology alone? Oxygen on Earth might be an example that gives us a toolkit. But what is a biosignature? Any detectable feature that must be produced by life or is most likely produced by life. This can be chemical, oxygen, methane, coexistence, organic molecules with unusual ratios, isotopic fluctuations. There could be structural biosignatures, stromalite, microfossils, layer sediments, atmospheric biosignatures, oxygen, methane, patterns that cannot be sustained by geology alone. Temporal biosignatures like life changes its environment with time, so the signature should vary in a way consistent with biology. Technosignatures, artificial signals, radio, gas, night lights. There's a big problem with false positive and false negative here. The philosophical challenge, we can find something that looks biological but isn't. For example, methane on Mars could be biological or rock water reaction. Oxygen on exoplanet could be biological produced by water photolysis. Organic molecules in meteorites could come from abiotic synthesis. That's why NASA uses the biosignature confidence letter. You don't just say life immediately, you climb through detection, validation, excluding geology, confirming context, and multi channel evidence until we the final step. The simplest explanation is biology. Sorry. How do we recognize alien life if it's nothing like Earth life? That's a challenging one. We search for general principles and not Earth's biology, like energy flow, complexity, information storage, disequilibrium, environmental impact. And we search these in oceans of Europa, plumes of Enceladus, methane lakes of Titan, exoplanet atmospheres, ancient rocks on Earth. If something is organizing chemical systems using energy and leaving a trace that geology cannot explain, we consider it a biosignature basically. But it's very arguable and challenging field what biosignature is and what life is. So this is tough. Jacques Monod once said, what is life? A simple question, but the answer is impossible to give. So, and there was also interesting idea on Carl Woese. believe life should be defined by information flow rather than molecules. They can be the same thing as well. Oh, but yeah, let's continue. Let's talk about synthetic biology and protocells, the origin of life subject that emphasizes vesicles, peptides, pores, XNA, mirror life. This has become very interesting in molecular scale, this subject, as these vesicles can grow, divide, encapsulate RNA, conduct chemistry, fatty acid proto cells, and they are best models for early life. And peptide pores and interactions and different ways of um, early life situations, and there are XNA, synthetic nucleic acids, and mirror life, how chirality works and how life determines which molecular structure it chooses. We can also discuss that. We will discuss those in the later in our series in Origin of Life and more scientifically. So, first time in history, humanity isn't just studying life, we are experimenting with what life could be as well, synthetically. And I know a couple professors actually that working on creating this synthetic life, and they are going pretty close to it. And with the membrane news, it's going to be even more in the future. First, artificial life we created, synthetic biology. Let's continue. We also mentioned protozoa research as well, synthetic genomes, GNA, mirror life, chirality. These are can be a good example. Sorry, there's XNA, for example, compared to them, what we synthetically make, and there's an example of chirality, basically a mirror vision of the molecule structure. It has really interesting implications, and which chosen is very interesting. But let's talk about the three pillars of modern astrobiology. So there's origin of life, life in our solar system, and life beyond our solar system. So origin of life would be the chemical evolution. How did life start? What chemistry preceded biology? Were minerals, hydrothermal vents, ultraviolet chemistry, or ice important? There's this is where protocell peptide and early metabolisms thrive. And then life in our solar systems for Mars, Europa, and Saturn and Titan. These pillars include cryovolcanoes, methane lakes, subsurface ocean, plum chemistry. And for life beyond our systems, we can check atmosphere, spectra, biosignatures, and technosignatures. We are entering an era where we can identify life without ever going there. So today's story is really the story of a scientific revolution, because before millennium, astrobiology was a hope, and after 2000, it became an engine, one that builds, launches, recreates, and rewrites. So we still have challenges like what life is, what biosignature is, but we are still the first generation in human history with the tools to actually answer the question, are we alone? And at this point, the universe is starting to look suspiciously full. So thank you for joining this lecture, and I'll see you in the next one.